So in this lesson, we're going to explore a really cool technique in modern genetics called cloning. You and I see clones all over the place, but they're more commonly known as twins. Clones are genetically identical, and according to your previous lessons in heredity, we know that the genes code for your traits. So clones don't only have identical genetics, they also have identical traits. The idea of artificial cloning has been around for some time now. Can you imagine selecting one perfect soldier and cloning them into an army of super soldiers that all have the same physical traits as the original super soldier? Or taking your strongest basketball team up against Michael Jordan, or two Michael Jordans? Or how about cloning a team of 5 Michael Jordans? But if this technology falls into the wrong hands, we could potentially have a multitude of problems. In 1997, the first mammal was cloned. The process that they used is actually quite simple. That's all you need is 3 adults. With these 3 adults, an egg is taken from the first adult. Now the egg is important because an egg has the potential to turn into any type of cell that it gets a signal for. But we don't want the egg's DNA because the DNA only contains half the number of chromosomes that we need to make a full organism. So that's why we unnucleate the egg. We take out the nucleus. The DNA that we do want comes from lamb number 2. So the DNA in lamb number 2 is from a somatic cell. We take that DNA and insert it into the unnucleated egg. In the process in creating Dolly, this was done by creating an electric pulse and fusing both the somatic cell with the egg cell into one. Now, we use a process called nuclear transfer, which basically means that we take one nucleus and we replace it with another. But the process isn't quite complete because the egg cell won't grow on its own, so we have to insert it within the womb of a surrogate mother. Once the lamb is born, which adult sheep do you think the clone will be of? Because DNA codes are physical traits, and the DNA that we obtained from sheep number 2 was inserted into the empty egg, we can determine that the lamb will be identical to sheep number 2. And that's the complete process that scientists used to clone Dolly, the first mammalian clone. So let's review. We first have to obtain an egg cell and remove its nucleus. Next, we take a somatic cell or a body cell and transfer that nucleus into the unnucleated egg. We then clone these cells with the new DNA. Once that single cell has turned into many cells, we have 2 options. These 2 options are called reproductive cloning and therapeutic cloning. Reproductive cloning involves taking the embryo and inserting it inside a surrogate mother to create an entirely new organism. Whereas therapeutic cloning means that if we take the embryonic stem cells and given the right signals, we can make the stem cells turn into a heart, blood cells or bone marrow, or even nerve tissue.